So today we are going to start talking about biotechnology. So what is biotechnology? So if you break that word apart, you have two different word chunks. You have bio and you have technology. So if you think about those two words separately, it helps give you the definition of this branch of science. So on the screen now, you see it says applying technology to DNA and proteins. So DNA and proteins are both molecules that we have talked about that living things need to live. And we are using these living organisms or their DNA to modify products for a specific use. So we are using technology to manipulate these little bits of DNA to make something that will help us make life better, or to develop vaccines that can improve our health. Another practical use of biotechnology is DNA profiling. So analyzing bits of DNA samples to maybe catch a bad guy. So you could find blood samples, hair samples, or little bits of skin left over at a crime scene, and you can match that to a suspect to see if somebody is guilty or if they are innocent. A process that is used to match DNA is called gel electrophoresis. And we are going to be doing a lab with this technology later this week. But it is a very simple process overall. And what you do is first, you have to make sure the DNA is cut with something called restriction enzymes. And those are going to cut the DNA at very specific places, and it ends up making DNA certain fragment sizes. And then number two, the DNA fragments are placed into the positive end of a gel, and then electric is just applied to it, which causes the DNA to move through the gel at different speeds according to how big the fragment sizes are. And then once the DNA is finished moving through the gel, you can look at the patterns that are left in the gel, and you can compare the DNA from a suspect, for example, with the DNA found at the crime scene. When you cut DNA with restriction enzymes, there can be two types of cuts that are made. So in the image on the screen now, you see the DNA has been cut and it's left with these overhanging nucleotides, and we call those sticky ends. Another type of cut would be if it is cut just straight across the middle, and that would be called a blunt cut. It's very straight. This image here on the far left shows you the setup of the electrophoresis chamber. This is very similar to what you'll see later this week when you do this lab. On the right, you see an example of what you might see after you have run your gel. So there are 4 different bands in this gel. And at the top, that's where you would have placed the DNA. And the DNA fragments will separate based on their size. And so after the gel runs and you look at the banding patterns, you can compare the DNA found from the crime scene with the 3 suspects that you have, and you can see which one matches. So if you were to look at those 3 suspects, who do you think would be guilty? If you said person number 2, you would be right. So if you can compare the bands from suspect 2 with the crime scene, they are an exact match, which means that person's DNA was found at the crime scene. They are very guilty. Okay, some more useful applications of biotechnology would be cloning, something you have all heard about. So just creating a copy of an organism. You can have animal cloning, or you can have reproductive cloning. So reproductive cloning, it says, results in the birth of a new organism. So this is not the only type of cloning. You can just have cloning of certain types of tissue that you need if you need a tissue transplant. Cloning does not have to be an entire organism, so it doesn't always have to be animal cloning. Cloning was first performed in the 1950s on frogs, And in 1996, you all probably heard about Dolly the sheep. Okay, this is when a full organism was reproduced and was pretty successful, although cloning still has some negative side effects. So Dolly still did not live the healthiest life compared to just a sheep that was born naturally. But in this picture here, you can see how Dolly the sheep was cloned. So you start by taking an egg cell from a donor sheep, and you have to remove the nucleus. So you're taking the DNA out of the cell. And then what you do is you take the donor cell and you insert that into the empty shell. So you're taking the original DNA out and you're sticking in the DNA that you want to clone. And so now that the new DNA is in there, the cell is going to reproduce, okay, and eventually it can be placed into a surrogate mom where the embryo will grow and develop and then eventually be born. And then it says at the very end there, the clone of the donor is born. So why do we clone? So we can create livestock with a desired set of traits. So maybe we want our cows to be bigger, we want pigs to be bigger. We want our livestock to look and act a certain way. We can create organisms that can produce needed medicine. So we can manipulate and clone bacteria to make a certain type of protein that is needed to make medicines. Okay, we can also restock endangered species. So it is possible that if a certain type of plant or animal becomes too scarce on Earth, we can try to clone them to preserve them so that they do not become extinct. Well, what are some of the concerns with cloning? Well, just like with Dolly the sheep, she was not as healthy as other sheep, and so they can have a shorter lifespan. Also, the cost of cloning is a lot, so it's expensive, and sometimes it can be inefficient. So it doesn't always work. You have to do multiple trials to get the outcome you want, and as you're going through the trials, you just spend more and more money. Another type of cloning is called therapeutic cloning, and this is using stem cells to cure diseases or injuries. So stem cells, if you're not familiar with that, these are just cells that have not become a certain type of cell yet. So they are not blood cells, nerve cells, heart cells, or stomach cells, or bone cells. They are just cells that have not become a fancy special type of cell yet. And so scientists can take these stem cells and they can apply different growth factors to them to make them turn into the type of cell that they need to work with. And so they can do that and multiply the cells in the lab setting through cloning, and they can get a certain type of tissue that they need for maybe a certain type of experiment, or maybe they're trying to help somebody be healthier because they didn't have healthy cells in the first place. Another type of technology you need to be very familiar with is recombinant DNA technology. So recombinant DNA. So you're recombining and piecing together different types of DNA to get a certain outcome. And these pieces of DNA are coming from two different sources. So I could take, in the example below, the gene from a jellyfish that causes it to glow. I can take the gene from a jellyfish and I can stick it into a regular fish, and now we have what we call glowfish. And you can buy these right now at a pet store if you wanted to have glowing pet fish. When you do this, this is just called genetic engineering. You are tinkering and manipulating DNA for a practical use, usually practical. Having glowfish probably isn't the most practical use, but we can use this to create medicines that we need to help treat certain types of disorders and diseases. Okay, the very last slide here just talks about genetically modified organisms, or you might hear that abbreviated as GMOs. So this is an application of recombinant DNA technology. You are just taking DNA from one organism and you're putting it into a different type of organism. And this just helps to create organisms that have a certain trait.